Usually, when we talk about a physical barrier, there's, a certain expectation of security.
Right, yeah.
Like a vault. You lock your valuables inside a heavy steel safe, spin the dial, and you just assume they are totally contained.
It's a necessary assumption for our peace of mind, really.
Yeah.
We rely so heavily on anatomical boundaries to tell us, where a pathology begins and where it ends.
But when you step into the world of early breast cancer, well, that solid steel vault often behaves a lot more like a flimsy screen door.
Exactly.
We're looking at a microenvironment where a completely contained preinvasive cluster of cells, this is ductal carcinoma in situ, or DCIS, somehow manages to just pick the lock of the local immune system.
It's wild. It breaches the duct, invades the surrounding breast tissue, and then transitions into invasive breast carcinoma, or IBC.
Right. And understanding the exact mechanics of that escape, like how it actually happens, has been one of the most frustrating bottlenecks in oncology.
Oh, absolutely. I mean, we've always known that invasive breast carcinoma presents with this profoundly suppressed immune microenvironment compared to DCIS.
Yeah, the cytotoxic response is just flattened.
Right. But we've essentially been showing up to the crime scene after the vault has already been blown wide open,
Yeah, without knowing the sequence of events that disabled the alarm system in the first place.
Exactly.
Okay, let's unpack this, because today, for you listening, our mission in this deep dive is to perform a meticulous detail-by-detail extraction of a massive 2026 transcriptomic study.
Yeah, published in Cancer Cell by Bowie and colleagues.
Right. And they mapped the cellular landscape of 72 human patients, covering hormone receptor positive, HER2 positive, and triple negative subtypes. Plus, they mirrored that data against outbred Sprague Dawley rat models.
And they didn't just find a loose correlation, they found the specific cellular lockpicker.
Which is incredible, but let's break down how they even saw this. How do scientists see what individual cells are doing in such a chaotic environment?
Well, the only way they could do that were by analyzing the tissue at an almost absurdly granular level, using single-cell RNA sequencing, or cRNA-seq.
Okay, so instead of looking at the whole tumor as a blended smoothie, they are pulling out every single piece of fruit.
That's a great way to put it, yeah. When you have a lesion transitioning from in situ to invasive, the bulk tissue data is incredibly noisy.
Right, it's just a mess of signals.
Exactly. So by utilizing cRNA-seq, they isolated the transcriptome, basically the active genetic construction manual of every individual cell across that evolutionary timeline.
But looking at millions of single-cell transcriptomes, that creates a massive high-dimensional data problem. How did they pinpoint the one specific cell driving the invasion?
By using specific algorithms to map it all out.
Mm-hmm.
First, they use something called UMP.
UMP, okay.
Yeah, which helps project that kind of multidimensional data into a visual map. Normally, you see distinct clusters, your epithelial cells here, your standard T cells over there.
Like perfectly separated islands.
Right. But to find the culprit, they used another algorithm called Milo. And Milo is an algorithm for neighborhood analysis.
Neighborhood analysis.
Yeah, so instead of forcing cells into rigid categories, Milo looked specifically at the fringes of those clusters. They were hunting for the shapeshifters, essentially.
Ah, the cells caught right in the middle of changing their identity.
Exactly. And that led them directly to the cycTreg.
The cycTreg. Okay, we need to deep dive into this specific cell.
Yes, it's a highly specific and quite rare population of cycling regulatory T cells.
Let's look at the exact molecular signature here, because a standard regulatory T cell, its job is to maintain homeostasis, right?
Right, it stops the immune system from attacking your own body.
But this cycTreg is different. It's expressing intermediate levels of FOXP3, which is a classic TREG marker, and intermediate levels of the IKZF2 and IKZF4 maturity markers.
Yes, but the kicker is that it is screamingly high in MKI67.
And MKI67, just to clarify, that's
the proliferation engine. That MKI67 expression is the critical piece. It indicates a cell that is actively, aggressively proliferating.
So if we visualize this for you listening, imagine the immune system's CD8 positive cytotoxic T cells are the police, right? They kill the bad guys.
Great analogy.
And regulatory T cells, the normal ones, are like internal affairs. They keep the police from overreacting.
Right.
But this newly discovered cell, the cycling TREG, with its MKI67 engine redlined, it's like a corrupt academy. It is actively mass-producing crooked internal affairs officers right at the boundary of the lesion.
That is a highly accurate way to visualize it. Their whole purpose is to completely shut down the police, letting the cancer escape.
Wow. And how do we know they are positioned right at that critical boundary?
Because of how they mapped the developmental trajectory. They used another algorithm called Monocle 3.
Okay, monocle 3.
Yeah, it performs what's called a pseudotime trajectory analysis. By inferring the lineage progression, Monocle 3 proved that these cycTregs are practically non-existent in healthy breast tissue. And they are present in very, very low frequencies, in pure indolent DCIS.
But right at the tipping point of invasion.
They explode. Up to 55% of all tumor-infiltrating lymphocytes in the invasive state are these cycTregs.
55%?
Yeah. They completely dominate the immune landscape at the exact moment the tumor breaks out of the duct.
That is unbelievable. Okay, so before we move on to the next piece of the puzzle, let me just bullet point this first part for you really quick.
Go for it.
Bullet 1. Researchers mapped the DCIS to invasive transition using strNA-seq and the Milo algorithm. Bullet 2. They found a specific proliferating cell, the cycTreg, which has that FOXP3 intermediate and MKI67 high signature right in the gap.
Perfect.
And bullet 3. These cycTregs act as a corrupt academy, suppressing the immune system to allow the tumor to invade.
Exactly.
So a question for you listening. Have you ever wondered why some precancers just sit there harmlessly for years while others aggressively invade? What if the difference is literally just the presence of this one corrupt academy cell?
It really reshapes our entire understanding of disease progression. But we have to remember, a regulatory T cell, even an intermediate one, doesn't just spontaneously decide to hyperproliferate in a vacuum.
Right.
It requires an activation signal.
Yeah, the corrupt academy needs a dispatcher handing out the replication orders. And I know the study looked into the myeloid compartment for this.
They did, specifically highlighting a CLEC10A high type 2 conventional dendritic cell, or CDC2.
Okay, but I have to push back here a bit. A CDC2 is a classic antigen-presenting cell.
It is, yeah.
Its primary role is supposed to be the informant. It shows the police what the cancer looks like to prime a robust anti-tumor response. It's not supposed to trigger a massive suppressive TREG expansion. So why are our own informants helping the cycTregs?
Well, you totally expect them to be priming the CD8-positive effector cells. But the tumor microenvironment is just remarkably adept at co-opting local myeloid cells.
Wow.
Yeah, in early DCIS, while standard macrophages were actually depleted, these specific CLEC10A high CDC2s were uniquely elevated.
Okay, so there are tons of them around.
Exactly. And they weren't just passively existing nearby. They were physically interacting with the cycTregs.
You're talking about the immune synapse, right? The physical connection.
Precisely. The researchers mapped it out perfectly. The CDC2 presents the antigen using HLA class 2.
Right.
But the actual proliferation trigger, the real order to multiply, comes from the costimulatory ligands. The CDC2 uses CD80 and CD86 to bind directly to the CD28 and CTLA4 receptors on the cycTreg.
So wait, the dendritic cell is literally grabbing the TREG and feeding it the proliferation orders through that physical CD80 to CD28 handshake.
Yes, exactly.
But how do we know for a fact that this specific physical interaction is what drives that massive MKI67 engine we talked about earlier?
They actually ran an elegant ex vivo assay to prove it. They used a fluorescent dye called CellTrace CSFE to track cell division.
Okay, how does that work?
Well, every time a cell divides, the dye concentration dilutes by exactly half in the daughter cells. So it allows you to count the exact number of generations a cell has gone through.
Oh, that's brilliant. It's like the ultimate biological paper trail.
It really is. So when they co-cultured T cells with normal peripheral dendritic cells, there was minimal proliferation.
Nothing crazy.
Right. But when they swapped in the tumor-derived CDC2s, the cycTregs rapidly pushed past what we call division 4. They were multiplying exponentially.
Wow.
And the definitive proof came when they added antibodies to block either the HLA-berry receptor or the CD80 ligand.
So they blocked the handshake.
Exactly. And the moment they prevented that physical handshake, the cycTregs completely stopped dividing.
That is just incredible. The tumor is literally reprogramming our own immune informants to train the enemy.
That's exactly what's happening.
Okay, time for our second quick recap for you listening. Bullet 1, CDC2s, specifically the CLEC10A high ones, are the accomplices. Bullet 2, they physically lock onto cycTregs via an immune synapse using HLA2 and CD80 to connect to CD28 and CTLA4. And bullet 3, the CSFE dye experiment proved that only tumor-conditioned CDC2s trigger this cycTreg multiplication. So my question for you listening, if the tumor is literally reprogramming our own communication lines to train the enemy, how do we cut those lines?
Well, cutting the lines is one thing. But even with an accomplice giving the orders, a hyper-proliferating cell requires a massive amount of metabolic support.
Right, it needs fuel. The immune cells alone can't sustain that kind of energy demand in a chaotic, hypoxic tumor microenvironment.
Exactly. There has to be a structural component feeding them.
Yeah.
And to find it, the researchers brought in another algorithm called CellChat.
CellChat. So we've got Milo, Monocle 3, and now CellChat.
Right. CellChat is an algorithm that infers cellular communication networks across the entire microenvironment, and it revealed that the dominant survival signals weren't coming from other immune cells at all.
Where were they coming from?
They were originating from the tumor's structural scaffolding, specifically inflammatory cancer-associated fibroblasts, or ICFs.
Fibroblasts. So the cells that normally just, build healthy connective tissue are being corrupted here too. Which specific variants are doing this?
They pinpointed ICCAS marked by high CD34 or high IL-6 expression.
Okay.
And the CellChat analysis showed that these specific ICKFs secrete a really potent cytokine called interleukin-33, or IL-33.
And let me guess, the cycTregs are perfectly adapted to catch it.
You guessed it. The cycTregs highly express a receptor called ST2, which is the specific receptor for IL-33.
So IL-33 is the fuel. Let's build out our analogy again. If the CDC2s are the dispatchers giving the replication orders, these ICCAFs are building a physical safe house right around the lesion.
That's a great way to look at it.
They are continuously pumping out IL-33 to provide the food and shelter that keeps the cycTregs alive, dividing and suppressing the good CD8-positive cops.
And we actually have spatial data proving just how literal that safe house concept really is.
Oh, really? How did they image that?
The researchers utilized multiplex ion beam imaging, or ME.
ME.
Yeah, ME allows for subcellular spatial resolution of dozens of proteins simultaneously, so they can see the exact coordinates of every single cell type in the intact tissue architecture.
Like a super high-res satellite map of the tumor.
Exactly. And they look at tissue from patients with pure DCIS, so patients whose lesions had not invaded yet.
Okay.
The MeBe spatial imaging revealed that a fibroblast activation, protein-positive CAFs, FAP-positive CAFs, were physically clustered in close proximity to the DCIS epithelium and the immune cells.
Yeah.
It directly predicted whether the patient would later suffer an invasive recurrence.
Wow. So the spatial mapping literally predicts the crime before it happens.
It does.
Okay, let me do our third bullet recap for you listening. Bullet 1.
Yeah.
The CellChat algorithm identified structural fibroblasts, the ICFs, as key communicators. Bullet 2. These ICTAFs secrete IL-33, which binds to the ST2 receptor on cycTregs to sustain them like a safe house.
Right.
And bullet 3. Using MeBe imaging, they saw that the physical proximity of these cells in early DCIS directly predicts a future invasive recurrence.
Perfectly summarized.
So think about your own neighborhood for a second. If you knew exactly where the criminal safe house was, wouldn't you target the house itself rather than just chasing individual bad guys in the street?
Which is exactly where the clinical translation of all this data becomes so incredibly compelling.
So let's bring this all home to the patient. We have the mechanism, we know the cells. How do we use this ecosystem, the cycTregs, the CDC2 accomplices, the iSafe safe house, to actually save lives?
Well, first we look at the large-scale human databases to understand the stakes. By analyzing the TCGA and Metabrick cohorts, the clinical implications become undeniable.
What did they see in the human data?
In early, grade 1 pure DCIS, having a high cycTreg score is the single, only immune subset associated with disease recurrence.
It's literally the canary in the coal mine.
Exactly. And once the disease progresses to the invasive stage in the Metabrick cohort, the mere presence of these cycTregs is the strongest predictor of poor disease-specific survival.
Because at that point, the cycTregs have completely dominated the landscape, right? They've caused a severe depletion of the CD8-positive effector cell diversity. The anti-tumor response has just flatlined.
Precisely. So to figure out how to dismantle this corrupted ecosystem, they shifted to a highly accurate in vivo model. They used NMU-induced Sprague-Dawley rats.
Okay, wait. Why Sprague-Dawley rats specifically? Why not standard lab mice?
It's a great question. Because Sprague-Dawley rats are outbred, their genetic diversity and their spontaneous tumor progression mimic the human immune microenvironment much, much more closely than standard inbred mouse models.
Okay. Well, here's where it gets really interesting for you listening. Because the researchers didn't just find one vulnerability in the system, they found two completely distinct therapeutic angles.
They did.
Let's look at therapy 1, dual targeting. The transcriptomic data showed that cycTregs highly express a receptor called OX40, or TNFRSF4. But I'm a little confused by their strategy here.
Why is that?
Well, they used an agonistic OX40 antibody in the rat trial. If OX40 is on a suppressive regulatory cell, wouldn't an agonist, like, turn it on more and enhance its suppressive function?
You would completely think so.
Yeah.
And historically, that's been a major concern. But context is everything here.
Okay, explain that.
In the setting of a highly proliferative, already hyperactive cycTreg, hitting it with an OX40 agonist pushes it into metabolic overdrive.
Oh.
Yeah, it actually destabilizes the cell. It forces the cycTreg to stop cycling and literally strips away its suppressive capacity.
That is a brilliant tactic. You're basically using its own hyperactive engine against it to induce exhaustion.
Exactly. And then they pair that agonist with a PD-L1 blockade.
Okay, so a 1-2 punch.
Yes. The OX40 agonist neutralizes the suppressive cycTregs, while the PD-L1 blockade simultaneously removes the inhibitory brakes from the CD8-positive cytotoxic T cells.
And what were the results in the rats?
The combination was remarkably effective. They saw a massive stabilization or complete regression in 75% of the treated tumors.
75%?
Yeah. The cycTreg population just plummeted, and the CD8-positive effector cells flooded right back into the tumor microenvironment.
So they completely reversed the immune suppression just by essentially overloading the cycTregs. But what about the second approach, up there too, going after the structural safe house, the IL-33?
That approach actually yielded some of the most profound systemic results in the entire study. They used a neutralizing antibody to block IL-33.
Right, so literally cutting off the primary fuel supply from the ICFs.
Yes, and by starving the cycTregs of IL-33, the tumor shrank, as you'd expect. But the fascinating part was what happened outside the local tumor microenvironment.
We need.
Starving the cycTregs triggered a massive systemic immune awakening. They observed a profound expansion of EOMS-positive memory effector CD8-positive T cells.
Okay, those EOMS-positive cells, those are the heavy hitters, right? They are the veteran effectors that have recognized the tumor antigen and are primed for long-term patrol.
Exactly, and they were patrolling everywhere. The researchers found these veteran effector cells circulating in the bone marrow, in distant lymph nodes, and even infiltrating distant completely healthy mammary glands.
Wow. So by simply cutting off the localized IL-33 fuel supply, they didn't just collapse the local tumor, they established a body-wide systemic surveillance network.
It's incredible.
Okay, final recap for you listening. Bullet 1. Human clinical data from TCGA and Metabric shows cycTreg levels strongly predict recurrence. Bullet 2. Targeting OX40 alongside PD-L1 destroys the cycTreg population by putting them into metabolic overdrive, shrinking tumors in the rat model.
Yep.
And bullet 3. Neutralizing the IL-33 safe house starves the cycTregs and boosts a body-wide EOMS-positive immune surveillance.
That's the whole picture.
So, question for the audience. We just went from mapping a single mysterious cell on a computer screen to literally shrinking tumors by cutting off its food supply. What stands out to you about how precise modern medicine has become?
It really represents a fundamental paradigm shift in how we approach early-stage interception. I mean, if we look at the totality of what Bowie and colleagues have mapped out here, we can clearly synthesize that the transition from localized DCIS to invasive carcinoma is not a random event.
Right.
It's not dictated solely by, accumulated genetic mutations in the epithelial cells.
It's an actively premeditated escape act.
Exactly. It is a highly choreographed, localized immunosuppressive program. It requires the cycTregs to proliferate. It requires the CDC2s to physically transmit the activation signals via that immune synapse. And it requires the ICAFs to construct an IL-33-rich safe house to sustain the whole operation.
So what does this all mean for you? Being well-informed in modern oncology means recognizing that we've really moved past the era of viewing cancer as just a rogue mutating cell.
Yeah, it is a corrupted ecosystem.
Understanding this ecosystem is the future of immuno-oncology. The future of intercepting cancer before it turns deadly isn't just about poisoning the epithelial cells. It's about systematically dismantling the cellular infrastructure that allows them to thrive.
And the precision with which we can now map and target that exact infrastructure is what makes these findings so incredibly actionable.
Absolutely. And I want to leave you with one final provocative thought to mull over.
Oh, let's hear it.
We started today talking about a locked vault, and we've spent this entire deep dive dissecting exactly how these cycTregs act as the ultimate lockpick in early breast cancer, bridging that gap between a harmless state and a deadly invasive one.
Right.
But we also highlighted that this exact mechanistic pathway is deeply conserved across entirely different species. It functions identically in both outbred rats and humans.
It does.
So, If this specific biological corrupt academy, the CDC2 accomplices, the cycTregs, the IL-33 safe house, if this is so universally hardwired into our immune architecture, could this exact same corrupt academy of cells be the hidden key that unlocks the invasion of other types of cancers in the body?
Oh, that's a brilliant question.
Could dismantling this exact same ecosystem be the key to stopping invasion in the lung or the colon or the pancreas? Keep that deeply conserved mechanism in the back of your mind the next time you read a headline about a new cancer breakthrough.